Hi, everybody. It's Lori Dameron from St. Martin Careers here at UNH. How are you guys doing? You're all a little bit muted, but I hope everything's going well for you. I wanted to thank you for your participation in this webinar. We have some really terrific representatives from Pfizer who will be facilitating the webinar. All of the presenters are going to talk about what's going on at Pfizer, more specifically in the Andover site, and they'll explain to you how to, kind of access opportunities. So our presenters are from Andover, but as you all likely know, Pfizer is a large multinational company, so there are many, many opportunities. And this is just a new arm of Pfizer that's being launched in Andover, Mass. So I am going to turn it over to Kelly, I believe, and Kelly's going to kind of take it from there.
Hi, everyone. My name is Kelly Savan. I'm a graduate of UNH of 1999, so quite a while away. I've worked at the Andover site when it was Genetics Institute, Wyeth, and now Pfizer for almost 21 years. And had a very successful career here, and I'm happy to talk about opportunities that we have coming up and to share a little bit about the Andover campus. I am the quality assurance lead on our plasmid project, our new suite that we're building here in Andover. So quality assurance, what we're responsible for is ultimately for release of the product for the next manufacturing. So we're overseeing all of the quality aspects of a product to ensure that it's safe, that it's done what we said it's going to do, and it's going to be the right thing to be able to give to patients. So we're making all of those decisions every day for our patients. So that's a little bit of what I do, and then introduce Jim, and Ravi and Laura can give their introductions as well.
Hi, good afternoon. My name's Jim McColgan. I am leading the operational readiness team at the Andover site as part of the startup of Pfizer's gene therapy effort in Andover. Kelly's gonna, and so I have three people from my leadership team here, Kelly, Laura, and Ravi. Both Kelly and Laura are UNH graduates. Alumni. We're going to go over a high-level overview of the Andover Pfizer, and then the Andover site, and then the gene therapy and the opportunities there. My own background, I'm a graduate of UMass Amherst, have a bachelor's and master's in microbiology, have about over 30 years of experience in the biotech industry, everything from bench scale up through very large scale commercial manufacturing. So very excited about the being in the gene therapy. It's a brand new and very exciting area. So I'm going to leave most of the talking to my leadership team, but I'll turn it over to Laura so she can introduce herself.
Hi, everybody. It's Laura Moore. It's great to speak with you all here today. So I am a UNH graduate as well. I guess as others have mentioned. So I graduated in 2006 with a bachelor's in microbiology, and then again with a master's in public health. And I've been working with Pfizer for about 15 years now almost. So I started right as I graduated and have moved through several different positions within the quality organization here at Pfizer. So right now I'm currently on Jim's leadership team, and I lead the quality control efforts. And quality control is essentially responsible for all the testing of all the products. So, we want to make sure, the only way to make sure that we know what we're putting out is, that we are, dispositioning what we say we're dispositioning, and testing is kind of the only way to really prove that. So QC in the labs, testing all the samples is super important. And so we're really excited to start the gene therapy program here, and learning a lot of new things through the process. So I will pass it to Ravi.
Hello, everybody. My name is Ravi Krishnapa. My background is in chemical engineering. I graduated from UMass Lowell with a master's in chemical engineering. I've been working with Pfizer for the last a little under 5 years. My current role in the plasmids team is that of a people leader. My job responsibility is working with technicians, process engineers, and my boss, the operations leads, to make sure that we produce gene therapy products consistent quality and quantity, and we meet the demand that's out there for the patients, and while making sure that our quality is of the maximum of what is we intend to produce to begin with.
Okay, Kelly, you want to take it, and you'll take it and go through the overview.
Yes. So as people have questions, please put them into the chat function. Laura will be moderating the chat, and we'll look for any questions you have, especially during the presentation, but certainly after the presentation, the floor is yours to ask questions of us. Okay, so I will share my screen now to go over a little bit about, Ravi and I will go over Pfizer and a little bit about gene therapy as well. Okay, who we are and what we do. All right. Okay, so first of all, Pfizer, number one is our patients. Our purpose, and we're all driven through this, the breakthroughs that change patients' lives. We look to this in everything that we do. Our decisions are around what is best for the patient and how we're going to help the patient population. This is really driving, as I said, the decisions that we make every day on how we make the smallest decision on how we document things to the largest decisions of what projects to move forward. So this is very important. And actually, I'll go over in a moment the leadership team, but we have a new CEO, well, not really new anymore, but new as of 2019, January 2019, and he really drove that new purpose for us. Okay, so our company, Pfizer, is quite large, as you may already know. We have significant revenue within 2019. We have 42 manufacturing sites worldwide, a number of them within the US, but within many countries across the world. And much of the work that we do, not only just gene therapy, but much of what we do with a number of our products within Andover and in other projects, and we're working with sites that are around the world as well, within the US and internationally too. So we're not just staying within our own bubble in Andover, but constantly interacting with those other manufacturing sites too. We sell products throughout the world internationally as well. We have a number of significant products that are substantial, not only in sales, but also in the population that they reach. And then not only that, but our clinical profile, which I'll go over in a little bit more detail, is quite extensive as well. So we are very firmly here. We've been here for, I think the original company was founded in the late 1800s, and we are here to stay for a long time. And of course, with all that work, we have a large number of colleagues that are around the world as well. And just at the Andover site, which the four of us are from, we have about 1,600 colleagues that are at the Andover site. Okay, this is the executive leadership team within Pfizer. As I said, Albert Bourla is our CEO as of January 2019, and has really looked at the culture within Pfizer and our purpose, and drove us to the purpose of breakthroughs that change patients' lives, but also taking a look at the values that we live every day, how we get work done, not just what type of work we get done, and then our bold moves. We're looking at those things. What are the key things that we need to do to drive our company forward and drive our products to our patients as easily as we can? Okay, so Pfizer's, there's a number of different departments, but the groups that we in Andover, and then really are doing the work of research and development, and then manufacturing, the WRD group that's within Andover across the world, and then what we call the Pfizer Global Supply, which is the manufacturing section of Pfizer. So we have a number of different pieces within the WRG department, or not department, but group: oncology, internal medicine, vaccines, information, immunology, rare disease. Now, a number of the products that we manufacture in Andover fall within each of these buckets, and we're very proud that reach many different types of patients as well. Okay, and as I said, the Pfizer Global Supply is our manufacturing operation. That is the group that we operate under, that Jim's team operates under, in bringing plasmids and the gene therapy business to life. It's under the PGS. That is a quite significant organization within Pfizer. We have, you can see here for the facts and figures of what we produce. The number of colleagues is quite significant. And again, all those manufacturing sites are within Pfizer Global Supply across the world. Okay, so our pipeline, as I mentioned, is quite extensive. If you are familiar with the clinical pipeline, how it goes from discovery to commercial, there are a number of different phases. You may hear of some of those phases when you look in the news about different products coming to market, what phase that's in. So from discovery to registration, there is phase 1, which as of January 28th, Pfizer had 26 products, possible products in that phase. And then phase 2, 37, phase 3, 23. And at any point in those, we're looking at different aspects of the product and how they can help patients. And are they helping patients? Are they working? So they're continually being evaluated and modified. And then in Andover, we're really looking at the products that are come to us for manufacturing around phase 2, phase 3. And then we are a launch facility within the Pfizer Global Supply Network. So in Andover, the products will come to us around phase 2, mostly phase 3, and then we'll supply the commercial market until it needs the product and perhaps is moved to another site for varying reasons. Okay, so not only that, we're here to help our patients, but we're here to, as a business, but we're here to help the patients in a different way too, and not just through the business. And Pfizer has a strong corporate responsibility to be able to help patients on this manner as well. We have several different partnerships throughout the world where we are helping organizations reach our medication, not only through purchasing, but also through these types of alliances as well. And one large one that we work with is called the Gavi Alliance, which supplies our Prevnar vaccine to babies around the world. Now, Prevnar, you may or may not know, is on the standard immunization schedule for babies within the U.S., and then is filed throughout the world. But the Gavi Alliance provides those vaccines to patients that cannot otherwise afford them as well. So we're very proud of these things that we're able to help the world in many different ways. Okay, so I'll hand it over to Ravi at this point to talk a little bit about the Andover Gene Therapy Project.
Thank you, Kelly. So how is Andover playing its role in the gene therapy? Before we get into that, let's take a few minutes to talk about gene therapy. What is gene therapy? So for individuals who have a genetic disorder, usually it's because of mutations at the genetic level at the genes within the DNA, which are sections of DNA. So how do we treat that? So in gene therapy, the whole concept is they use a nucleic drug delivery system to repair and replace the mutated or ineffective gene. So how to do that? In order to do that, they use what is known as a vector. In our case, the vector we are going to be using is an adeno-associated virus. As you all know, a virus is a machine at a microscopic level that is very, very good at infecting human cells or any cells or the cell of a host cell, and then replicating within the host cell rapidly. So we use that to our advantage, and we genetically engineer the virus so that unbeknownst to the virus, when it does affect a host cell, it injects the gene of interest, also known as the transgene, that we have genetically inserted so that it can go then, and when it takes over the enzymatic machinery or the cellular machinery, it injects this transgene or the gene of interest into the patient, thereby giving the patient the ability to produce proteins that the gene prior to the treatment was unable to produce. So we can talk a lot about this, but in a nutshell, this is exactly what it does. We are using a virus to deliver the payload of our medicine at the cellular level. So gene therapy has been in the talks for the last 2 decades or so. There was a spike in interest in around the late 1990s, but there was a death of a patient in one of the earlier clinical trials around 2000 or 1999 that kind of put a hold, and FDA became kind of put a hold on any further development of this this avenue of drug therapy. But moving forward, there has been a lot of progress in vector engineering, and a few early successful trials led to a spike, as you can see, around the late 2000s for many indications such as hemophilia, muscular dystrophy, genetic blindness, and sickle cell disease. And more often than not, these therapies were more effective when you considered mutations within the cell because of a single gene. And they were more effective, and most of the studies right now are focusing on in that respect. For example, in our Andover, we are focused on, Kelly, next slide. We're focused on developing a gene therapy for Duchenne's muscular dystrophy. Duchenne's muscular dystrophy happens because of a single mutation or a mutation in a single gene within the human body that is responsible for the production of a protein called dystrophin. Dystrophin is a protein that a normal human being would require, and it makes effortlessly so that there's proper and normal development of muscular and skeletal muscles. However, with patients afflicted with Duchenne's muscular dystrophy, because of the mutation in the gene, they do not produce the requisite amount of dystrophin, leading to a muscular degeneration and weakness over time. It's termed as a rare disease, but it's pretty common at 5,000 males at birth. It's more common in males than females. Typically, in the past, the patients would die young, but because of better palliative care these days and advances in cardiac and respiratory care, they are kind of extending the life expectancy so that they can they live a little longer. But nevertheless, when you consider the average population, people with DMD die in their early age. And this is the project that gene therapy project that Pfizer is working on to produce a genetic drug for the Duchenne's muscular dystrophy. So how do we do that? So what we do is, in essence, we use a host cell called E. coli. So as you know, bacteria has 2 kinds of chromosomes, 2 kinds of DNA. One is a nuclear DNA that's within the nucleus of a bacterial cell. Its main job is to give the cells certain traits that it passes on from one offspring to another, so that it can thrive and survive. Whereas there is another set of DNA called the plasmids, which is outside the nucleus, and this is a special DNA that gives the plasmid special characteristics to survive harsh conditions. For example, it may give the bacteria ability to survive antibiotics. So plasmids have these unique characteristics that impart characteristics to the bacterial cell to survive under unpleasant conditions. So what we do is we use these plasmids and we genetically engineer the plasmid DNA, which is a circular DNA, snip a piece of the DNA and insert the gene of interest. For example, in this case, for Duchenne's muscular dystrophy, we would insert a gene that would have the ability to produce dystrophin. And this gene is called, this transgene is then inserted back into the E. coli. And then we use this E. coli as mini factories, so to speak. And as the E. coli reproduce and populate, each of these E. colis, unbeknownst to them, have the plasmid within them that has the gene of interest. So we use these as factories, we harvest them, and then we take these plasmids out of them that have the gene of interest. This plasmid that we produce in Andover is then termed the critical starting material, which is then shipped to another facility at Pfizer, where they take this plasmid and then package it into a virus, and then use the virus, the adenoacetate virus, as a vector, so that can be then injected into a patient, and then the virus then infects the host cell, and lo and behold, unbeknownst to itself, the plasmid transgene is inside the cell, giving the patient the ability to fight off the inability to produce the gene dystrophin that, the protein dystrophin that they were not able to produce. So in leading to this effort, we have a manufacturing facility that we are building as we speak. It is expected to go online sometime early next year. So between now and then, we plan to staff up our operational staff and then enabling functions to about 55 people. Currently, we're at 12, so we need another 40 to 45, 43 or so. Of these other roles, there are about 35 of them that specifically fall under my boss, Tim Coyle's group, the operations lead. And then some under enabling functions such as quality control and quality assurance, supply chain, and so forth. We have positions for both first shift and second shift. We are looking at people who basically have a science background or an engineering background, sciences being the life sciences and engineering being chemical engineering or biomedical engineering. We are hiring in a staggered fashion. We just closed out a round of open positions that we posted out. It closed. Our next round of positions would be opening up in end of May and June. So if you go to Pfizer.com right now, you may not find any postings, but pretty soon you should have postings for both process technicians and process engineers and technicians. Eventually, we will be hiring other positions such as in the leadership roles, such as people leaders and technical leads. But for now, pretty soon you should be seeing postings for process engineers and technicians. So a little bit about our facility. So the plasmid facility is built from the ground up, but then it's built from inside out as well, because a current existing facility, there was a facility called the Andover manufacturing facility, which was decommissioned, but it lies within the entrails of the building A. So they carved out space within, they decommissioned the facility and they carved out space from within it. So it's been built inside out. As you can see the 3D renditions at the bottom of your screen, that's how it would look. And that's a high level overview of the pink regions are the ones that belong to ACMF, that belong to plasmid manufacturing facility that's been carved out from inside surrounding existing manufacturing facilities. So our pace of hiring or timeline, so to speak, so we are right now in May, we are looking to hire between now and end of December, close to 55 to 60 people. And we intend to keep up this pace, and we are reaching out to people and universities and organizations to hire talented people. That's why we are here today, to give you an overview and spike your interest and encourage you to apply. This is a picture of our facility. It's right off of 93. At the bottom of the picture, you would see the highway that you see is 93. So it's a 70-acre site. It's one of the few facilities that has both research and development, clinical facility, manufacturing facility, and a commercial production facility all on the same site. We have about 1,800 colleagues on site. Kelly spoke to you earlier about a strong pipeline that we have at the clinical trials. Our building E, and our clinical manufacturing facility, which is the white building to the north of your picture, is where we're producing our drugs for clinical manufacturing facilities. And then the building to the left of the pond are buildings A and B, where we are actually doing the commercial productions. And our plasmid manufacturing facility is deep inside that building B, from the inside out, that's been built from the inside out. So a little bit high-level overview on what we produce at Andover. So currently, Andover produces about 6 commercial products. For example, Benefix. It's a product that is basically a protein for factor 9. People with hemophilia who don't have the natural ability to produce the protein have a problem with blood clotting of blood. Benefix is a supplement that people take. That's one. It's a rare disease that we are producing a drug for that. Infused bone graft, also known as bone morphogenetic protein, is another drug that we produce. It's a drug that's kind of absorbed in a sponge-like gel, and then they can use that gel as a scaffolding and wrap around the bone, and it promotes bone growth. In case of, like, for example, people with maxillofacial surgeries or having developed with bone issues, this bone is kind of the sponge is kind of biocompatible, and it absorbs the BMP protein from there and promotes bone growth. Kelly spoke to you earlier about Prevnar 13. It's our Prevnar vaccine. Every single person, baby in the United States, when mostly all of the students here probably had it as a kid, over 1 billion doses, and pretty much Andover is one of every single one of those billion doses that's out there had its start in building A at Andover. It treats pneumococcal diseases, and it's a very, very popular drug for the rest of from Pfizer. It's a flagship drug, so to speak. The next one is Besponsa. Besponsa is used to treat lymphoblastic leukemia. It's a MAB, monoclonal antibody. It's a more recently commercially approved drug. In addition to, and this was one of the new drugs here, in addition to bevacizumab, which like your generic drugs in your pharmaceuticals, this is a biosimilar to one of the drugs that's already out there, and it treats. It has several indications for cancers such as colon cancer, ovarian, cervical cancer. Another drug is adalimumab, which is a biosimilar for Humira that you may have seen ads on TV. It used to treat rheumatoid arthritis, Crohn's disease, and spondylitis, and many other drugs. So these are the drugs that are produced right now that are already commercially approved. But as Kelly suggested, we have a strong pipeline, many clinical trial candidates, in addition to what the gene therapy candidates that we are actually in clinical trials right now. So if you have any further interest in products or positions, strongly encourage you to go to Pfizer.com. Make sure you chose North America and then Andover, and you should be able to see the positions that are there. LinkedIn is another example to patch up with the alumni who are working in Pfizer. That's a good start. Alumni is always a good starting place to learn more about your interests versus what's out there at any of the Pfizer locations. And we should open up the floor to any questions. You can take the questions as they come up.
Thank you, Kelly and Ravi. So I'm monitoring the chat room. So if anybody wants to, you can type into the chat and I can read the question, or you're also more than welcome to unmute yourself and ask a question directly if you'd like to. I just request that you try to remember to mute yourself afterwards, just so we don't have too much feedback.
So I do actually have a quick question. One, thank you guys so much for doing this. It's really awesome to learn about everything that you guys are doing. I know you said that the next round of openings is going to be for like end of May, early June. If I've already applied to a process, I believe, technician position within Pfizer, it's probably not within this program, right? Would it be able to transfer it to this program? Because this is something that I think is really, really interesting. Or is it kind of all under the same umbrella?
Yeah, go ahead, Jim.
Yeah, so I can take that. So if you applied for a process technician, maybe a process scientist. So in addition to the process engineering positions, there's also process scientist positions. If you applied for that in one of the other suites, then we would not necessarily get that resume unless the hiring manager shared it, but it wouldn't come to us directly. So rule of thumb, you can't over apply for a job. So I would encourage you to apply again. Now, the hiring managers do tend to share candidates, but there's no guarantee.
Yeah, and just to add to that, even if they've shared resumes, you need to formally apply for a position to be considered too.
So may I just add to that? I know working with students, they're a little reluctant to apply to more than one, to an employer because they're concerned about the appearance. And I just want to make sure everybody understands that it's entirely appropriate to apply to more than one job within a given employer, particularly if it's a larger employer such as Pfizer. So if you guys want to speak further to that, that would be really great. Yeah,
because we get hundreds of resumes, and then they use a sorting algorithm. And to be honest, we don't totally understand the sorting. So it doesn't count if you apply to multiple positions. If you apply to multiple positions you're not qualified for, that's much more of an issue than applying for multiple positions for which you are qualified for. For example, we'll have positions that clearly call for a science or an engineering degree, and we'll have people applying for multiple slots in those positions that don't have any of the basic requirements. And our hands are tied. You know, if we publish that you need a bachelor's in science, then that's what's required.
Okay, so we have a question coming through the chat. Thanks again for holding this. As you are hiring over the next few months, are you planning on onboarding large groups at once, or are you planning on having people start more gradually as you hire?
Ravi, you want to take the time frame question?
Yeah, I can give you an example. Like, for example, this recent round of postings that closed, we advertised for process technicians and process engineers, different grades, for about 15 people. We were looking for about 6 process engineers and about 9 technicians, three technicians for each of the categories. Now, in an ideal world, we would find 15 qualified candidates, in which case we would say, hey, those postings are completely filled in, so let's go on to the next set of requirements, right? If we don't find, we may have to repost. So you may see one title process engineer, one position, but it may have different grades, like grade 1, grade 2, and grade 3. The difference being that some may be entry level, some may be with a bachelor's degree, some may be with bachelor's plus master's and a few years experience. So we typically are posting for multiple positions, different grades at any given time. The progress forward all depends on how, what kind of a quota of resumes we get and how we evaluate them and how we hire them. So yeah, in short, we do tend to hire, post at least batches of requirements, batches of positions.
And I'll just add to that, obviously, in the current environment, we're trying to figure out how we onboard and train people. I mean, my whole team, we've all been working remotely for the last 2 months. So we do have a hiring. If you think of that graph that Ravi showed, shows the hiring cadence. We're trying to balance the available candidates, how many we can onboard and train at a time, as well as when we need people on board. So we're not doing one at a time, but we would tend not to bring on 15 all at once, you know, on a single week, just from a logistics point of view. And just to add something to Ravi, we're looking for process techs, senior process techs, which tend to generally have a minimum of an associate's and a bachelor's, as well as process engineers requiring an engineering degrees, and process scientists who requires a life science degree. So micro, biochem, biology, something like that. So what the environment we work on is very much cross-functional with both life sciences and engineering together.
Okay, so we have another question. Interested in R&D, what specific types of positions will be opening up in this next round?
Again, I can answer that. So these are not R&D positions. These are in Pfizer global manufacturing or global supply. So when I'm thinking R&D, and I started my career in R&D, if you're interested in being at the bench and, you know, in a lab type setting, running experiments, you know, at the, 10 mil, at the Eppendorf tube level scale, 1 mil, 10 mil, 2 liters, those in R&D, those positions are posted in Pfizer, and there are a number of them available in Andover, but they're just not in this project.
And so I'm going to try to lump some of these together. So will there be any part-time or temporary positions available? And I'm going to include that. Included with that is internships opening. Yeah.
So internships, it's a very sore subject, and unfortunately Pfizer had to cancel its internship this summer, as many companies had to. There were very few intern type positions that are amenable to remote working, and in the current environment we just didn't feel comfortable bringing in students to do summer internships. Right now, we do not have any part-time or we do not have any part-time positions. We do have some temporary positions. Unfortunately, we do not hire directly. We contract through various temp companies for temporary positions. So we don't advertise, but we will, in addition to those 60 odd positions Ravi went over, there's probably another 10 to 20 of temp positions in both our group as well as some of the enabling functions. Our temp positions will start onboarding in the fall. But you can't, we don't advertise them directly. We have various service providers, which you can apply to them and get your resume in with them, but we don't hire directly. And that's just the Pfizer policy.
Yeah, and so for Ravi, if you could explain a little more about the roles for, say, process technician, engineer, or a chemical engineer. So different roles and responsibilities that one could expect.
Okay, I'll give you a snip into what a typical day is. For example, we typically operate on a shift basis. So when you start your shift, a people leader or a supervisor gathers in a huddle room with the process technicians and the process engineer, and we go over the list of activities that we need to do on that and any given day in any given shift. And that's mainly geared towards producing drugs and meeting the supply and demand equation, right? So you go into so you have a list of activities. We have different areas of operations, such as upstream operations, downstream operations, upstream being cell culture, downstream being purification. We have glass wash and autoclave areas. And then we have our product services where they actually formulate all the medias in the buffer. So we all split off into groups, taking the list of activities that we need to do. For example, formulate buffer X, formulate media Y, inoculate the fermentation vessel, conduct ultrafiltration, diafiltration operations, or chromatography step. So in this activities or subset of activities, what the technicians would do is they have standard operating procedures that tell them how to operate a certain task, how to execute a certain task. And they are the ones who are executing the task. They make sure they execute the documentation for the SOPs. They interact with the automation to make sure all the recipes are run, so that recipe A is run in respect to how it's been prescribed. What a process engineer does is he assists that entire technicians in making sure that all of these activities are done in what is known as a right first time scenario, right? If there's a glitch in the automation, the process engineer would then contact the automations people to make sure that it's resolved in a timely manner, so that we can meet the demands of the day. We can produce what is necessary to be produced in the day. If there is an SOP or a standard operating procedure that on the floor is leading to ambiguity for a process technician, the process engineer would make sure that he would take that SOP and do what is known as a CI, or a continuous improvement, or what is known as or he would tweak the format so that any person should be able to understand that. So all of those right first time activities, process troubleshooting, process monitoring, data acquisitions, batch record reviews, all of that is done by the process engineers, so that at the end of the day, when the shift is done, the end goal is that as a team, teamwork is vital in Pfizer. No one person can achieve our goal of achieving breakthroughs for patients on a daily basis. It is a team effort. So between the process technicians, the engineers, and somebody like a supervisor like me, who would make sure that at the end of the day, we have checked off our boxes on, hey, we did all of these activities right first time, no impact to product quality, no investigations, no deviations, no safety incidents. As a group, that's what we aim to do on any given basis. And as a chemical engineer, you would fit that role where you would have to look at avenues where you can improve the process, look at avenues to save resources such as time, or how look at innovative ways of how to do a certain documentation or how to do a certain step, cut out waste. Those are the things that we would look for an engineer as a perspective of how you would look from outside and how to achieve that end goal as a team.
Kelly or Ravi, Kelly or Laura, you want to add anything to that in terms? Kelly, I think you're mute.
You're on mute.
All right. So I was just going to add that, as we said, gene therapy, plasma manufacturing is really on the leading edge right now. And so what Ravi described is an ideal situation, but we know that there's going to be a lot of opportunities for troubleshooting and learning, and for a lot of us, not just the new hires, but all of us that are on the team. So I think we anticipate that there'll be a lot of opportunities for learning, and probably some things that may go wrong, and learn and grow from those too. So it's not going to be as simple,
Yeah.
I would say.
Yeah,
Actually,
I think that's really important to point out too, and it also sort of, it makes it exciting for those of us who like the challenges and working through things that we don't necessarily know, and I'm not sure how to articulate it, but yeah.
I just want to
Totally left me.
Thanks, Kelly, for pointing out, because the picture I was trying to paint is applicable when you're in a state of control, when your process, your facility, and your documentation is in a state of control. And given that we are having the construction going on till the end of the year, and we'll be stepping into a brand new facility, brand new process, brand new equipment, we will not be in a state of control. We can be rest assured of that. We will have a lot of issues with automation equipment, and that's where, as a team, we will have to do corrective actions and preventive actions and remedial measures on the go, so that we take the facility that's brand new to a state of control where we can then produce drugs for the commercial supply.
Yeah, so I would just add a couple. First of all, I know Ravi knows this. We will be in a state of control. Otherwise, we won't be running. It's maintaining that state of control that is actually critical. When you think about what we do in biotechnology and in biopharmaceuticals, it's really complex, and we kind of lose sight of it because we deal with it every day. There's a huge amount of troubleshooting. We're dealing with biological processes. We're dealing with cells who we have one idea what we want them to do, and they've got their own ideas on what they want to do. It's a constant troubleshooting. My team will often hear me say that I really would look forward to, I really want a month of boredom. I really want a month of everything, no excitement, and we're lucky if we get a day or two. So on the flip side, it's an extremely dynamic environment we work in. It's also very much a collaborative environment. It's very much, as I was saying before, and as Ravi was saying, it's very much in a team base, because we need engineers and biologists and scientists working together. We need to have a fundamental understanding of what's going on outside in terms of heat transfer, mass transfer, KLAs, the stuff that engineers are trained for. We deal with very large pieces of equipment, very large bioreactors, very large volumes of fluid moving around. At the same time, we need to have equal expertise in the biology of what's going on. As we look at metabolic byproducts, how much lactate, ammonia is in the media, what does that mean in terms of metabolism? What's happening in glycolysis, the TCA cycle, electron transport? Stability of molecules by being bound by bonds and ionic strength of solutions is critical as we move through that. So it's very much a team-based effort where you bring the expertise of multiple disciplines together. And in terms of a state of control, if you think everything we make are getting injected into people, and most of them are sick because you're not taking our products if you're healthy, other than our vaccines. And that's even a higher burden, because you're injecting a perfectly healthy child, usually a baby, with a vaccine that doesn't have a disease to present a disease. So the focus on making sure that molecule and what we're making is perfect is extremely high, and it can be very daunting, but it can be very rewarding as well.
Yeah, and I think I just want to point out too that in the gene therapy space, it's new for all of us, and this is a really exciting time, because to be able to come in and work in a space where not only is the product new, it's a new facility. So there's a lot of it's not often when you can have an opportunity to have this experience to work with a new facility alongside cutting edge science. So this is sort of like a once in a lifetime type of opportunity in the therapy space.
Yes, and it's extremely, that's a good point. So when this facility is complete, it will be, if not the largest, one of the largest facilities for making bacterial plasmids as part of gene therapies in the world. We're going to be taking a process that has been moving extremely fast, because when you think of the patient population, they have a limited amount of time that we can get to them due to their disease. We're taking a process, we're going to be running it at a scale that has never been run before, significantly higher, in types of equipment that's never been run before. We need to, in a brand new facility that nothing has run in before. Any of those challenges individually would be daunting. Putting all three of them together is unprecedented. So it's very exciting. It can also be very daunting, which is why when we're looking for talent, we're looking for people, not only with the academic background, but the teamwork, the ability to think outside the box, the collaborative skills that will enable us to do that.
Yeah, thank you.
Laura, I'll take this last question because I know it's a difficult one.
Yeah.
Yeah. So it's, I was wondering if international students under the OPT will also be considered, and if yes, does Pfizer have sponsorship for them after the OPT? If this, I'm not quite sure with OPT, I think what it is, if this in terms of sponsoring for a visa, like an H1, H10 visa, unfortunately, Pfizer will not do that for entry-level positions. And that's totally out of our hands. So one of the first things is going to be a requirement is that anybody has to already have the appropriate paperwork to work in the United States. And I know that's a difficult message, but unfortunately, it's the reality that we're living with right now. So hopefully that answered that question.
Yeah, thank you. So another question came in around, I had the opportunity last summer to shadow a couple program directors and principal investigators at NIH. Are there any shadowing opportunities in the gene therapy project over the summer? Thank you. So we kind of touched upon this earlier too with the internship program, and Pfizer has a fantastic internship program. Typically, due to the COVID situation right now, and most of us are working from home and trying to keep essential worker only on site. So unfortunately, they've had to cancel the internship program for this year. Hopefully we can pick it back up at some point in the coming years, but for the program at this point, there are no job shadowing or internship type positions.
Yeah, unfortunately not, because we probably, myself and my team probably won't be back on site until towards the end of the summer full time, right? The facility's under construction. What we're doing is very much paper-based, electronic, documentation, regulatory. We don't really need to be back on site until the facility's at a state of completion where Rave and his team can get in there and we can all get in there and start doing stuff. And that's going to probably be September. But job shadowing opportunities, if it's flexible, that's an excellent example. You use your network out there to try to do that. And if it's unpaid, even better, because there's much less paperwork to do. If you say you want to come in for a few days, that's what you really want to exercise your network. And don't be shy about using your network. So if you have friends whose parents are in the industry, or they know somebody in the industry, don't be shy. I mean, let us say no, we can't do it, as opposed to people self-censoring themselves out.
So that's all the questions for now. If anybody has anything else, feel free to either speak up or write something in the group chat.
If I could just add too, so we're here to talk about the direct positions within the plasmid or gene therapy project at Andover, but there are always a number of positions that are open at the Andover site through the Pfizer careers website. So definitely take a look at those, whether they're in R&D, whether they're in quality control, whether they're in operations. You really can't go wrong starting at any area, because I will say that when I started, I'm not sure where I wanted my career to go, whether it was to stay in biotech or eventually I was going to go to the CDC, my original plan, but here I am, and I'm happy. So you just don't know, and it's good to just start in a department that is of interest to you, and you just never know where it's going to take you in your career. Yeah. And I just encourage you not to limit yourself in any of those departments that you may apply for.
Yeah, that's a really good point, Kelly. And I didn't know you could have been working for Anthony Fauci right now. You know,
right.
You know? Don't shy away from second shift positions. Most people on first shift ended up there by starting on second shift. Also, second shift, you get a shift differential, fairly significant at times, because we know it's a non-ideal shift. So don't do that. Don't shy away from that. Don't shy away from temporary and contract positions. That's essentially, if you're on a 6 month or a year-long contract, that's a 6 month or a year-long job interview. And we try to hire contractors when their terms up, if we have the positions. There's no guarantee there'll be a position there. The other one is in these type sessions, don't get discouraged. You know, getting your first job, we all had to go through our first job. And to be honest, you guys have it much harder, even outside the current environment. It can be daunting. And I have my daughter's a recent college graduate, and I have a son in college, and I know the trouble she went through getting her first job. So the current round of, we got many more resumes than we have positions. So the vast majority of the people that apply that are qualified probably aren't going to get positions here. So doesn't mean you don't apply again. Doesn't mean don't restrict yourself to Pfizer, even though we'd like to have you people at Pfizer. We know we can't hire everybody. If you want to be in the biotech, biopharmaceutical industry, this is the best place in the country to be. In the Boston area, probably the second best is San Francisco. It keeps going back and forth, which is the most promising. But there are literally 200 companies in Cambridge and in the greater Boston area. Now, knowing this is UNH, most of these are probably up in New Hampshire, you know, a lot of people do that commute. You know, both Kelly and Laura work in Andover, but they both live in New Hampshire. So I would just say hang in there and don't restrict yourself, and stay positive. It is a difficult process we've all gone through getting that first job.
And I would strongly encourage you to reach out to your alumni. If they are in the Boston area and if they're working there, they're your best bet. Yeah. Reach out to them. And my other point would be is try to get any experience with any hands-on experience that you have at your college level, at your laboratories, or your professors doing a research project. Get that experience. It's a lot more. It gives me a lot more perspective and interest. And from a recruiter perspective or even an employer perspective, it's good to see hands-on, even if it's lab scale experience for somebody's resume, in addition to classes.
Yeah, and I would say for our positions, lab-based experience will be, we don't expect you to have experience of what we're asking you to do. A bioreactor will cost $1 million. They don't let undergraduates work on $1 million pieces of equipment. The purification that we do, very expensive pieces of equipment, very expensive reagents that we use. So, but having experience in a lab in an internship gives you ideally a leg up, because you have some awareness of being in that type of environment, which is very much similar to the type of environment we have in the PGS environment. So don't be discouraged that you don't have bioreactor experience or large scale chromatography or large scale filtration or any of those, because very, very few people coming out of any program would have that. But we're looking for the foundational elements that will allow us to build on that.
Yeah, so and to point out, to add to that too, so I think that's a really important point, is that when you're looking at the job descriptions and seeing what the typical tasks or activities that are expected, as new graduates, you're not expected to have full experience in all of those. We have a really strong training program and you will be trained for every task that you have to do. And so what's really important is sort of the foundational skills, that you have some sort of background and knowledge of, working knowledge of, putting liquids together, or, measuring, and, some of the basic techniques and philosophies there. And then just sort of that ability for absorbing information, for training and teamwork and collaboration. And so to that point too, just to not get too discouraged when you're looking for positions, and just keep applying. Apply.
Yeah, leverage your network, be on LinkedIn. A lot of these positions will be linked to people's profiles. And so that's another way to get information and awareness. So you see any more questions, Laura?
I don't know.
Okay. I don't know if Laurie or Emily, you want to have any last words, or how we will wrap up?
Sure. Well, I wanted to first just start by thanking Kelly, Ravi, Laura, and Jim for hosting this webinar. The questions were great. We have a lot of very ambitious, well-qualified students at UNH, as Laura and Kelly know. You guys are on the alumni team. You get a great education. So I wanted to thank our students for attending this as well. I hope it was helpful, and encourage you guys to apply and just get out there and put yourself out there. So a big thank you to all of you at Pfizer. And students, certainly keep your eyes peeled on Handshake and also on the Pfizer website. And do know that St. Martin Careers and Career and Professional Success at UNH are open over the summer, and we're always willing to give you a hand if you need it. So keep in touch with us as well.
I would just do not apply through Handshake. We're not allowed to take applications via Handshake by company policy. So I know we've done this and people will apply through Handshake, but please don't. We can't do anything with those.
All right. Great. Thank you.
Thank you.
All right. Thank you.
Thank you, Lori. Thank you, Emily.
Thanks.
Bye-bye.